In our last class, we have studied about some basics of classification, and today we are going to study uh, the classification given by R.H. Whitaker, and we will study today two kingdom. First is Monera, and second is Protista. So let's begin with it. In earlier classification, as you know, it was divided between two organism, plant and animal. But by the discovery of cell and then further discoveries of their components, there was further classification of living organism to the prokaryotes, eukaryotes, and all. So what are the basics of the classifications between the organism? The first one is the cell wall, if whether the cell wall is present or not. If it is present, what kind of cell wall does it have? Then comes to the nutrition, what kind of nutrition they have and how they fulfill their needs. What were the problems with earlier classifications? It was that prokaryotes and eukaryotes were placed together, unicellular organism and multicellular were placed together. For example, Cladomonas was a unicellular organism, while Spirogyra, that is an algae, is a multicellular organism. Both heterotrophs and autotrophs were placed in the same class because plants and fungi, they are two different kingdom. One is autotrophic and one is heterotrophic. The cell wall of fungi is made up of chitin, and the cell wall of plants is made up of cellulose. After the revolutionary step of five kingdom classification given by R.H. Whitaker, fungi was separated from the plant due to distinct nutrition, that is heterotrophic nutrition and the cell wall composition. So the question can be asked in the exam: what is the difference between the mode of nutrition in plant and animal, plant and fungi, and what is the difference between the cell wall composition of fungi and plant? Two different kingdoms were created. Both consist unicellular organism. Or you can say single cell, but they are divided further. The unicellular protist consists of Cladomonas, Chlorella, amoeba, paramecium. Significance of the change: the classification criteria shifted not just the morphology but physiology. So morphology and physiology, but also made the nutrition, reproduction, cell structure and evolutionary relationship इन सबको स्टडी किया. Now comes the phylogenetic. It is reflects evolutionary history. Further change will be better understanding. If we study our history, we will make sure our future will be brighter than before. The first kingdom we come across is kingdom Monera, that is you can also say bacteria. In kingdom Monera, all the members of this kingdom are all bacteria, none other than that. Ubiquitous means found everywhere. It is found in mountain, river, ponds, deep undersea. It can be found anywhere, everywhere. then present in extreme habitat. Uh, there are two characters. For example, there is a certain uh, hydrophile and which hates the water. They can grow in hot spring, they can grow in deserts. Snow, deep oceans can be free living or parasitic, can occur as both. And it is the one of the most abundant, not one of the, it is the most abundant microorganism on the earth. Classification by shape, they are classified into four types. First is coccus, plural cocci. Second is bacillus. Coccus are generally spherical shape. Bacillus are rod shape, or you can say cylindrical shape with the rounded ends and cylinder like structure in the middle. Structure, the simple cell structure of prokaryote, complex behavior and metabolically diverse. Metabolic diversity may have flagella and both nutrition in bacteria. Coccus are spherical shape, bacillus are rod shape, vibrio comma shape, and spiralum are spring-like spiral shape. The structure is they are simple single cell or prokaryotic. Prokaryote, what do you mean by prokaryote? The cells which don't have nucleus or cell organelles, but they also have, they may or may not have flagella. Flagella is for the movement of body. They can also have spore. What kind of nutrition is seen in the bacteria? There can be two type of nutrition. One is heterotropic and another one is autotropic. In autotropic bacteria, the mode of synthesis is photosynthesis and chemosynthesis. So what do we mean by photosynthetic or chemosynthetic autotrophs? These are the organisms which these are based on the what kind of source they use as energy for the process of autotropism. If they use sunlight to, then and this, it will be called photosynthetic autotroph. And if they use chemical substance like nitrifying bacteria, they oxidize inorganic compound, they will be called chemosynthetic autotroph. Now in general, the bacteria, or you can say Monera, are divided into two phylums. One is archaebacteria and otherwise eubacteria. First we will discuss about archaebacteria. Archaebacteria are the prokaryotes which are extremophiles. Extremophiles are those organisms which survive under very harsh, which can survive under very harsh condition, extremes beyond the stream, beyond normal. They are prokaryotic unicellular organisms and they have a unique cell wall. They have peptidoglycan on their cell wall instead of polysaccharide. Polysaccharides are the sugar like cellulose. They lack peptidoglycan. They have polysaccharide plus proteins or lipids. The plasma lipids have branch chain. The plasma membrane lipids have branch chains, resistance to the extreme conditions, which help them to survive in harsh weather. They are basically classified into three major parts: methanogen, halophiles, and acidophiles. Methanogens are the organisms which can survive in the marshy area where methane is produced. Uh, One of the example is methanobacterium. The halophiles are those bacteria which can survive in really salty conditions, or you can say seawater. They are called halophiles. And thermoacidophiles, which can survive in high temperature as well as acidic nature. They are found in hot spring near volcanic eruption also. The example are thermoplasma and sulfolobus. Methanogen are used in producing biogas. Halophiles enzymes used in biotechnology. And for thermoacidophiles, the enzyme are used in PCR. PCR is polymerase chain reaction, which is help in the replication of DNA, amplification of DNA. Since they can work on high temperature, they are used in as a tag polymerase in PCR process. What are the general characters of eubacteria? Eubacteria are also known as true bacteria. They are called true bacteria because they possess a rigid cell wall and motile they are also found in motile forms. The major groups are cyanobacteria and chemosynthetic cyanobacteria. They consist of chlorophyll A, like green plant. They are photosynthetic autotrophs. They are unicellular. They may be unicellular, conolient, or filamentous. The one of the example of filamentous state is nostoc. We will discuss it. They can be found in all kind of regions: freshwater, marine, and terrestrial habitat. Their colonies are surrounded by gelatinous sheath. As you can see, this is a structure of nostoc. There is another outer layer preventing the direct contact to the environment. This mucilaginous sheath protects the bacteria and also provide a sticky surface to attach sticky texture to attach to the surface. Some have special cell called heterocysts. Heterocysts are the cell which are used in nitrogen fixation. Chemosynthetic autotrophic bacteria. These are the bacteria which oxidize inorganic compounds like nitrate, nitrite, and ammonia to release energy used for ATP production. They also play an important role in nutrition recycling, like nitrogen, phosphorus, iron, and sulfur. What are the importance of these bacteria? They help in nitrogen fixation, for example, NAB9 and nostoc. Photosynthesis adds oxygen to the environment. Decomposer, they are good decomposer. They can recycle the dead organism into the inorganic matter, so that there is no less nutrient on the in the soil. Biochemical cycle: nitrogen, sulfur, and phosphorus cycle. Heterotrophic bacteria, they are most abundant in nature. Ecological role: they act as decomposers, recycling nutrients. Beneficial: they are used for making curd. Example, lactobacillus. Antibiotic production: streptomyces. Nitrogen fixation in legume rhizobium. They can be harmful pathogen too, which can cause disease in human, animals, and plants. For humans, the disease are cholera, spread by the uh, bacterium Vibrio cholera, typhoid, Salmonella typhi, tetanus, and citrus canker. This disease can be found in the plants related to citrus family like lemon, orange, etc. Reproduction in bacteria. Bacteria can reproduce by sexual and asexual both methods. Under unfavorable conditions, they form endospore resistant structure. There can be sexual reproduction seen in uh, in bacterium, conjugation, transformation, and transduction. They are basically sharing of genetic material or plasmid between the two bacterium. Mycoplasma, mycoplasma like organism. It's similar to this PPLO, pleuropneumonia like organism. These organism does not have cell wall, hence they are resistant to many antibiotics like penicillin. Penicillin is one of the antibiotic which acts on the walls of cell walls of the bacteria and penetrate it, and which can easily kill the bacteria because they don't have cell wall. The penicillin will not affect on these bacteria. They can survive without oxygen. They are facultative and anaerobe. What do you mean by facultative and anaerobe? They are aerobic, but in case they do not have oxygen, they will shift to the anaerobic respiration. Many are pathogenic to plants and animal. And I guess that's enough for today. Sorry for the inconvenience. We will continue our class for the topic Protista tomorrow. Thanks for listening. Have a nice day. Hope you like the video, and kindly share, like, and share, subscribe the channel.